Hello again, everyone. This is Cassandra Miller, and now I will be telling you about our other project, the impact of cell shape on the doses delivered to the nucleus from lutetium-177 labeled radiotracers. Now, if you're familiar with radiopharmaceutical therapies and dosimetry, you're aware that organ and voxel level dosimetry, often with PET and SPECT imaging, is the primary method of quantifying these therapies. And dosimetry has a lot of very beneficial uses, such as determining absorbed doses to tumors and organs at risk, as well as determining injected activities for future cycles of therapy. And this is very important and useful. However, dosimetry on this macroscopic level omits the physics and biology that occur on the microscopic or cellular level. There are microscopic variations in absorbed doses between radioisotopes and even between radiopharmaceuticals that cannot be seen at the organ level. For example, various properties such as the track length and even energy deposition patterns of different particles, i.e. alpha particles, beta particles, even gamma particles, etc., differ. Furthermore, different pharmaceuticals also have different uptake patterns within cells themselves, and all of these properties will not be able to be seen on the macroscopic level. Therefore, cellular dosimetry is key to enhancing our understanding of the mechanisms of radiopharmaceutical therapy. Cells in the body have many differences, and the one that I'll be looking at today is the shape of the cell. The shape of a cell depends on the organ, as well as the disease if you're looking at cancer cells. For example, in the purple image on the left, we have clear cell kidney cancer cells, and as you can see, they're approximately spherical in shape. They're not perfect spheres, but I would say that spheres are a relatively good approximation for the cell. And then in the gray image on the right, we have LNCap prostate cancer cells, and as you can see, they're not really spherical at all. They're much more elongated, not quite an oval, but elongated. We also observe when looking at LNCap prostate cancer cells on a plastic well, they're elongated as well. The shape of the cell may impact the distribution of the radiopharmaceutical within the cell itself, and it therefore might also impact the cell dose and potentially even the treatment efficacy, so it's something that should definitely be considered. Now, at this time, many studies at the cellular level are performed using simulations, and simulations require a definition of cell geometry. Spherical and oval models are common and often used because of their simplicity. However, for the reasons discussed prior, it's possible that a one-size-fits-all model may not be suitable for all cells or all cell types. Now, the aim of this work is to simulate 3 different LNCap prostate cancer cell models filled with activity of lutetium-177 and compare the doses per decay, or the S values, to the cell nuclei. Overall, the aim of this work is to see how the shape of the cell impacts the nucleus doses. Alright, so now I will tell you a little bit more about the methods behind this experiment. So GATE version 9 Monte Carlo software was used for all of the simulations. We based the cell areas on cross-sectional areas of human LNCap cells. So all of the nuclei and cytoplasms in the models that I will show you shortly have identical cross-sectional areas, and we kept the total cell volumes approximately constant at around 1300 micrometers cubed. Lutetium-177 was placed homogeneously in the cytoplasm or membrane of the cell, and we simulated activity in these cell regions separately. Now, because we are simulating LNCap cells, we are trying to emulate a PSMA-binding pharmaceutical, which we know has primary uptake in the cytoplasm or membrane, and that is why we made that decision. We simulated 1 times 10 to the 8 primaries, or 1 times 10 to the 8 decays of lutetium-177 for each model and cell region. And finally, to determine the S values, the energy deposited in each nucleus was scored on a voxel level using GATE, our voxel size being 0.179 micrometers cubed, and we converted this value to absorbed dose. Alright, so now I will tell you a little bit about my cell models. Firstly, I will mention all of our models had a nucleus, a nuclear membrane, a cytoplasm, and a cell membrane, and both of the membranes were 5 nanometers thick, although the exact dimensions of the nucleus and cytoplasm varied amongst the models. So for our first model, we had a spherical cell, which was just a spherical nucleus, evenly surrounded by both of the membranes and the cytoplasm. So our next model is an egg cell. As you can see, it somewhat looks like an egg, and this model has a flat bottom, and the reason for that is we are trying to simulate a cell that is attached to a plastic well plate, similar to the plate shown in this image here. And because of that, we can validate our results using ex vivo experiments. So in this model, the cell is much more elongated, similar to the LNCap cells we saw at the beginning, and the nucleus is a half sphere. For our final cell model, this was more of an oval cell, again to represent an adherent cell, so it was flat. And this model is somewhat similar to the egg cell. It is elongated, although now instead of a half sphere, the nucleus is a oval. To calculate the S value, we summed the dose deposited in each voxel I of the nucleus dose map of each model separately for the cytoplasm and the membrane, and we divided this value by the number of simulated decays, which as mentioned was 1 times 10 to the 8. Alright, so here are our results. This is a plot here of the S value to the nucleus in the 3 different cell models, from activity in the cytoplasm, which is in pink, and activity in the membrane, which is in green. So immediately, as you can see, the doses to the nucleus of the spherical cell are much larger than the doses to the nuclei of the 2 elongated models. And this is sort of what we expected, because the spherical nucleus is surrounded evenly on all sides by the cytoplasmic membrane, whereas in the 2 elongated models, the cytoplasmic membrane are somewhat more farther away from the nucleus, so this makes sense. We notice a slight difference between the nucleus doses within the elongated models, but the difference is not that large. And finally, we notice that the difference between the S values from activity in the cytoplasm and activity in the membrane is much larger within the spherical cell nucleus as opposed to the 2 elongated models. So what we found in this study is there are in fact differences in nucleus doses between spherical and elongated cells. We found a much higher dose to the nucleus of the spherical cell, and this tells us that cell shape may indeed be important, and it should definitely be considered during cell modeling and simulation studies. The use of an accurate model may result in incorrect results. And finally, to better understand individual therapy response to radiopharmaceutical therapy, a better understanding of dosimetry at the cellular level needs to be gained. There's a lot of very interesting information to learn at this level that could be applied to radiopharmaceutical therapies. The next steps in this work include comparing different radioisotopes and pharmaceuticals, as well as expanding this to a layer of cells and tissues, as opposed to just looking at a singular cell. Thank you everybody for listening. Please feel free to contact me if you have any questions.